And our next speaker is Diamantis Sellis from the LBB in Lyon, and he will talk about the evolution of IESs in Paramecium.
Okay, well, thanks for sticking out. I'm very excited to talk to you about some interesting new results in the evolution of internal eliminated sequences in Paramecium. We already heard many things about Paramecium life cycle and micronucleus, but just make sure that everybody's on the same page regarding what I'm going to talk about next. I want to remind you that one of the interesting and peculiar things in Ciliates is that the fact that they have two different types of nuclei, and I'm particularly interested here in the elements that are being removed from the micronucleus to create in the developmental process that creates the macronucleus. So these are the IESs which we find in large tens of thousands of copies throughout the genome, breaking genes. But the cell is able to precisely excise them in a TA boundary with a weak motif. These sequences are unique. They're not very similar to each other, so perhaps they evolve very fast. And they have a very interesting multimodal length distribution. Most of them are really short, around 27 base pairs, and then every 10 base pairs there's another peak in their size distribution, which is shown here. This has been hypothesized that it has to do with the mode of excision from the genome, but still there are many unknown questions. So, for example, we don't know where IESs come from. There is a hypothesis that they are relics of ancient transposable elements that the cell managed to get rid of. So, since they don't cause much trouble, they can still be there. There are many unknown questions regarding the molecular mechanism of the precise excision, how they can be targeted so effectively. Tens of thousands of IESs interrupting proteins are being excised and the cell is able to function. And also we don't know much about the dynamics of IES evolution through the long evolutionary history, how they appear, spread, and how they get lost from species. So in order to explore all of these questions, a consortium has been formed, led by Eric Meyer and Sandra Duacourt, which has been struggling to isolate and sequence both the micronuclear and the macronuclear genomes, assemble and annotate the IESs, and finally do the comparative genomic analysis, which I'm mostly going to talk about today. The group of Paramecium in this study are all part of the Paramecium Aurelia species complex. They are morphologically identical but genetically distinct. And also I'm also including Paramecium caudatum and tetrahymena, which are more quite distantly related species. Briefly, the methodology is simple. After doing all of the work of reading the genomes, the first step is to reconstruct the phylogeny of the group, because we had some previous attempts, but with just a handful of genes, there's not much you can do now. Since we have all of the genome, I believe we can do a much better job. Then the second step is to reconstruct the history of each single IS when it was inserted by going back in time. And now for reconstructing the species tree phylogeny, I'm using two approaches which largely agree with each other. Just to save some time, I'm going to show you one. Here on the one side, I'm showing a specific gene family for which we know which nodes corresponds to duplication, which node corresponds to a speciation event. And on the other side, we have the species tree, which recapitulates some of the findings of previous research and things we expected. For example, the Pentarellia, Primarelia, closely related ones, Octorellia, Tetrarelia. The only difference between the two approaches I used is the placement of, in the topology of the tree, and here I'm just showing topology, not branch lengths. The only difference in the topology is the placement of Paramecium Tridecarelia, which in the reconciliation, in this approach, is placed as an outside sister group to the set of these Aurelias, while in the previous approach is found to be a sister group to Pentarelia and Primarelia. Now, having this basis, which is the history, the history of each gene family for all the species, there are a number of downstream analyses we can do. I'm here only focusing on the ISs, so just to describe the picture of how ISs look in these species. We have more or less the same number of tens of thousands per each genome. A few exceptions is the Paramecium caudatum, which has a lower density, and Paramecium sonoberni, which has a much higher density of ISs. And all of them show the same really impressive multimodal distribution of length sizes. So here, in order to compress much information, I have a log scale on the y-axis, the number of ISs, and log scale on the x-axis, the length in terms of base pairs. With black lines is the Paramecium caudatum, the outgroup. It seems to have slightly shorter, it has about the same number of short ISs as the rest, and slightly less of the long ISs, but we can see the same periodicity. And most of the other species show what we knew already from Paramecium tetrarelia. They have the same more or less number and ratio for different length sizes. Two interesting exceptions are Paramecium tridecarelia and Paramecium sonoborni that show a very tight peak at 230, approximately 230 base pairs. I'm going to get back to this point later on. Now I'm going back into the method. So we have the phylogeny, we have of the species, we have the history of each gene family, which we constructed at the protein level. So this is what I'm showing up here. It's a protein alignment of one, just like a random sample. And then we can go back from the protein alignment, we can go back into the nucleotide alignment. And here, with black dots, I mark the positions in which an IS was present. So this is the macronuclear genome, where the ISs have been excised, so they are actually functional and they can create functional proteins. And I just mark with black dots the places where an IS was present and it was excised. And looking across different species only in these conserved regions, we can be confident that these ISs that are located in this located in this specific locus are homologous. So they were probably present in the ancestor of these two genes, either paralogous genes or orthologous from a speciation event. So once we have this presence and absence information, we can recode it as a presence-absence matrix with just zeros or ones or question marks if we don't know. And then we use a Bayesian framework to infer what was the ancestral state at each speciation node. And what I'm showing here is the species tree topology with the inferred rates of gain in each branch. The model of the inference we used assumes a constant rate of insertion. But even though we assume this, we see that at some particular branches the rate is much, much higher than other branches, which means that there was a wave of insertions that happened at this particular moment. And this seems to be just before the split after the split of caudatum from the rest of the Paramecium Aurelia group, where a number of whole genome duplications happened. And then there's a secondary wave that was on the branch that led to sonoborni with a much higher rate. We can do the same for the rate of loss of ISs, and again we see that the differences are maybe less pronounced, but still there are some clades with a higher rate of loss, leading to Pentarelia and Primarelia, and I'm not sure why these particular clades have a higher loss. That's something we still have to figure out. Now, since we have all of this information, we can add on this species tree all sorts of information that are particular, all sorts of particular properties of ISs. So here in this tree, I'm plotting the length distribution of ISs that were inserted at this node. So at this time point, so let's take this as an example. This is the same double logarithmic plot with the number of ISs and the length. So we see all the ISs that were inserted at this time, that were inferred to be inserted at this time point. have or expect their known length distribution. So there are many short ones and fewer long ones. While on the contrary, the ISs that were inferred to be inserted in only in Paramecium area, so very recently, they have a more flat distribution, more uniform. So this fits with our idea that they are inserted and they are probably long, and as time goes by, they disintegrate and become smaller and smaller. One interesting thing to notice here is in the Paramecium sonneborni clade, there is a peak of that corresponds to the 231 base pairs that I showed you before. These seem to be recent insertion of ISs only in this clade. These are about 500 copies that have a narrow length distribution, very centered around 231 nucleotides. They are very similar to each other, about 40% pairwise identity, which is very different from all the other ISs, which you can't even align them. They're so diverged from each other. And as a result, if we try and do some sort of phylogeny, they have a star-like phylogeny. They have terminal inverted repeat, which is similar to the motif of all ISs as we know, but it extends up to approximately 10 base pairs long. And they have the target duplication, the TAs. So all of these make us think that they look very similar to some sort of a mite transposon that did insert in this clade at some point in time, producing multiple copies and then became an IS. So this is quite exciting because it's the first time that we can actually see this process. So putting everything together is we inferred the phylogenetic history of Paramecium, and we reconstructed a detailed history of the evolution of all ISs. And now this enables us to see that, in fact, we had at least two waves of IS insertions, one after divergence from Caudatum and one on the branch leading to Paramecium sonneborni, and that there is a correlation between length and age. Recent ISs are longer on average. And another rather unexpected surprise is that there have been quite a lot of recent complete losses of ISs in specific clades, for which we still don't know what is the molecular mechanism. Finally, I would like to thank lots of people that were part of this project, that started from actually culturing, isolating the nuclei, sequencing, doing all the identifications, annotations, developing methods specifically to deal with these complicated inference problems, and you for listening.
Okay, I have a question. I was wondering if the rates of insertion or deletion of ISs that you observe, do they correlate with any other features of a genome, like, for example, the gene retention and loss rate? Because, I mean, I noticed that the lineage going to Paramecium sexaurea had a pretty high rate of new ISs, of gains. And sexaurea is the one that has been losing its gene very fast. So, you know, I was thinking maybe these guys have low effective population size, selection is not very efficient there. So did you look at that?
I haven't made correlations yet, but I think the gene content of all species is about the same, so I wouldn't expect many large differences. But it's something to look into.
Sexaurea has like a couple thousand genes fewer, less than like, for example, by Aurelia.
Oh, so then that's a big effect.
There might be like some signal to see that. We have time for another question.
Diamantis, it's very tempting to relate the wave of insertions that you had after the divergence with Caudatum with the whole genome duplications that occurred at that point. So wondering whether the whole genome duplications can provide an opportunity for more IS insertions, in fact, then we would wonder whether something similar happened in the branch that leads to sonneborni, for instance. And so my question is, can you actually, is there anything to be seen in the last whole genome duplication from the Sonborni species? And can you actually be sure that this duplication is the same, at least, that occurred in Sex Aurelia? Can you recognize the two homologs from the last whole genome duplications when you compare Sex Aurelia and Sonborni?
So the question is if there was an effect of, I'm not sure. So you're asking if the fact that we had a whole genome duplication creates somehow a wave of insertion?
It could lower the selective pressure against new IES insertions, because now you have two copies of a gene, so it doesn't matter so much if you lose one, right?
Yeah.
So you can imagine whole genome duplication as a formidable occasion for IESs to insert, or for transposon sequences to insert and be transformed in IESs softly to recover the function of the gene later. So then this peak that you have in the branch leading to Sonborni is suggesting, was there something, I mean, did the genome get duplicated independently from the other Aurelia species?
I don't know. I don't know if there was an event of whole genome duplication before Sex Aurelia and Sonborni. And if that was...
So we don't... it's pretty clear there's not an additional duplication in Sex Aurelia. Sonborni we don't know yet. But I think we can keep talking about that during the coffee break. Because we're running a bit out of time, so I'm sorry, we're going to have to... Thank you. And,